everyone, Gauri here. Today's video is on polymerase chain reaction PCR, which is a method widely used to rapidly make millions to billions of copies of a specific DNA sample. Let's begin. In 1983, a technique was conceived by Carey Mullis of Cetus Corporation that has become widely used to amplify specific DNA fragments without the need for bacterial cells. This technique is known as polymerase chain reaction or PCR. The most popular use of the thermal cycler, sometimes referred to as a thermocycler, PCR machine or DNA amplifier, is to amplify DNA segments using the polymerase chain reaction. The procedure constitutes three major steps, which are denaturation, annealing, and elongation. There are various components which are necessary to perform PCR. The basic procedure used in PCR is depicted in the following slides. The technique employs a heat-stable DNA polymerase called Taq polymerase that was originally isolated from Thermus aquaticus, a bacterium that lives in hot springs at temperatures above 90 degrees Celsius. In the simplest protocol, a sample of DNA is mixed with an aliquot of Taq polymerase and all 4 deoxyribonucleotides along with a large excess of 2 short synthetic DNA fragments that are complementary to DNA sequences at the 3' ends of the region of the DNA to be amplified. The oligonucleotides serve as primers to which nucleotides are added during the following replication steps. The mixture is then heated to about 95 degrees Celsius, which is hot enough to cause the DNA molecules in the sample to separate into their two component strands. The mixture is then cooled to about 60 degrees Celsius to allow the primers to hybridize to the strands of the target DNA, and the temperature is raised to about 72 degrees Celsius to allow the thermophilic polymerase to add nucleotides to the 3' end of the primers. As the polymerase extends the primer, it selectively copies the target DNA, forming new complementary DNA strands. The temperature is raised once again, causing the newly formed and the original DNA strands to separate from each other. The sample is then cooled to allow the synthetic primers in the mixture to bind once again to the target DNA, which is now present at twice the original amount. This cycle is repeated over and over again, each time doubling the amount of the specific region of DNA that is flanked by the bound primers. Billions of copies of this one specific region can be generated in just a few hours using a thermal cycler that automatically changes the temperature of the reaction mixture to allow each step in the cycle to take place. To check whether the PCR successfully generated the anticipated DNA target region, agarose gel electrophoresis may be employed for size separation of the PCR products. The size of the PCR products is determined by comparison with a DNA ladder, a molecular weight marker which contains DNA fragments of known sizes, which runs on the gel alongside the PCR products. That is it for today. I hope you found this video on PCR educative. Thank you.